CM Bio is a UK-based startup company, and we develop bioengineered human organ-on-chips technologies. LiverChip is a smartphone-sized device that models the structure and function of the human liver by incorporating a scaffold which holds human liver cells into this device which mimics the circulation of the blood flow through the human organ. And we connect this device to various sensors so that when the liver becomes infected in the model, and then we use drugs to remove or treat the infection, we can measure those effects in real time. So the cost of developing drugs is extraordinarily high for pharmaceutical companies. Current estimates are around $1 billion and more than 10 years of development to get new drugs to market. And the cost of developing new drugs is very substantially driven by obviously clinical trials, but also the requirement to do extensive testing in animals before humans can take new medicines. So if we were able to use technologies like LiverChip or like our hepatitis B technology, Quantum B, we might be able to substantially reduce the cost of developing these new medicines by reducing or eliminating animal testing. A basic experiment using LiverChip can start at around $22,000 as compared with animal testing, which can range anywhere from $50,000 to more than $1 million for extensive long-term safety or carcinogenicity testing.
Now, this might sound more like something from the script of a sci-fi movie. The futuristically named Quantum B is a chip that unbelievably contains cells that represent a tiny human liver. Scientists here in the UK have developed this new technology to help speed up the discovery of new drugs to treat diseases such as hepatitis B. It could also help to reduce the number of animals used in drug testing. Well, for more on this, I'm joined by Health Check presenter and family doctor Ayan Panja and Dr. Emma Skeets, who works at CM Bio, the company developing these human organs on a chip. Thank you both so much for joining us. Dr. Skeets, I know you've got it in your hand, so can you just show us and just tell us a little bit about it?
Sure. So Quantum B, this is a device around the size of a smartphone. What we do is we take the cells from a human liver and we introduce them into a specially designed chip that is built into the device. We don't recreate the entire organ on the plate. What we do is take the smallest number of liver cells, which together will function like the entire human organ. And you can see that on this plate, we have around 12 individual units that we can use for testing drugs.
Wow, and how much of a breakthrough is this?
Yeah, the breakthrough is quite significant. So to date, researchers have only been able to perform research, animal research on hepatitis B using chimpanzees. And for obvious ethical reasons, many drug companies have almost completely stopped using chimpanzees in drug testing. So to be able to evaluate new drugs before they go into the clinic and before they're given to patients using a technology like Quantum B can really help speed up and reduce the cost of developing new medicines.
Ayan, why is it so important, to find some kind of a solution to hepatitis B?
Well, a number of reasons. Firstly, it's a viral infection that's extremely infectious. And secondly, there's no cure for it. Most of the treatments we have at the moment are antiviral drugs or interferon, which is a type of signalling protein. But people with the virus are at increased risk of developing liver cancer and definitely an early death. So it looks pretty bleak. And in terms of drug research, there's really nowhere to go. And this could revolutionise that. So it's really exciting.
Is finding a cure for hepatitis B the only way to combat the disease?
Well, there are other things. I mean, someone who has the disease, certainly vaccination is the big thing. So in the UK, we see very little hepatitis B because we have a vaccine that's readily available. But in the developing world, that's a real issue. In terms of staying healthy, someone who's infected ought to consider, reducing their intake of alcohol and living a generally healthy life, and they must make sure that they get a flu jab if they can. But really, there is no cure, and you can only maintain that disease for so long. So this gives us real hope.
Ayan's just pointed out that this is a big problem in the developing world, hepatitis B. So how will this work, and will you be able to use it in some of these countries?
So to be very clear, the Quantum B is not actually a cure for hepatitis B. More what it does is it speeds up our ability to find these new medicines. We're working with members of the scientific community and drug companies using this technology to speed up the search for a cure. And that's really how this technology will have an impact for patients, is bringing new drugs to market much more quickly.
So what's the future for this chip? I mean, is it likely to get smaller? This is the first model, I suppose.
So smaller is better in terms of this technology, because it means that we can have more of these types of chips on a plate. Smaller can also be useful because we can join a number of these chips together to form like a human body on a chip. So we're part of an American-led program which is researching body-on-a-chip technologies. And we can use these types of devices to see how the effect of a drug which is intended to help one organ, such as the heart, could potentially have damaging effects on another organ, like the liver. And so we can use technologies like the body-on-a-chip technology to avoid those kind of problems as we move through the drug discovery process. And of course, this can really help to reduce the number of animals that are used in drug testing, and much reduce the risks for volunteers who participate in clinical trials.
Dr. Skeets, Ayan, thank you both for joining us, and very fascinating stuff there. Thank you.
The ability of scientists to develop new drugs is hampered by the fact that testing on animals has limitations when it comes to use in humans, whilst testing in humans has the potential for dangerous side effects. But new technologies being developed around the world are now replicating human organs on chips, allowing scientists to test drugs in a safe and controlled way. I'm Dr. Goody Singh in London, England, looking at the potential for this type of bioengineering to improve the way that life-saving drugs are made.
Organs on chips replicate the tissue structures and biological function of human organs, and those being developed include lungs, hearts, kidneys, and livers. This then allows researchers a quicker and easier way to test new drugs for some of the biggest global diseases. Just outside London are CN Bio Innovations, an organisation who have created a human liver on a chip with the hope that new drugs can be found to help treat and cure liver diseases.
So the thing that's really different about this device is that we try to actually mimic the microarchitecture of the organ. This is a polymer disc, and it's got 300 small channels. The dimensions are such that when we organise liver cells into these channels, they form these sort of long donut-shaped arrangements of cells. And this is really trying to model the architecture of the liver, which is a very vascular organ. And
so there's lots of blood vessels.
Exactly, lots of blood vessels, and essentially your cells are arranged around these blood vessels. And then this device, this plate, you put 12 of these chips into the plate, and the plate enables us to flow the fluid containing various nutrients and oxygen past the cells to keep them healthy and functioning.
So the fluid simulates blood then?
Exactly.
So is that like having 12 miniature livers?
Exactly. There are 12 little organs that we can actually perform testing on. And once the chips have been inserted, the plate will then be ready for us to introduce the liver cells for the experiments.
One of the key advantages of the liver chip is that because it mimics the structure of the liver so well, the liver cells stay alive for several weeks. This means that the scientists can study in detail the impact of a new drug on the organ.
Many of the diseases of the liver or the toxic effects of drugs on the liver only build up over a number of days or weeks or months. And so it's difficult to observe those types of effects or predict those types of effects if you're using a technology where the cells are only functional for a matter of hours.
After introducing a drug to the liver chip, the team can study the impact it has by performing the lab equivalent of a biopsy or a blood test. This allows them to test whether the drug is toxic or to see how successful the drug might be at treating disease. Reducing the dependency on animal testing enables Emma and her team to run a huge number of tests, gathering large quantities of data much more quickly before clinical trials begin.
So the test that we're doing here is it's like the equivalent of a biopsy, is that right, in terms of looking at a sample of tissue from the liver?
Yes, so Emma's taken one of the liver chips out of the plate in the lab, and you can see that within the scaffold there is a regular array of channels, and these are filled with liver cells. So in fact, the good news here is that the drug has had no negative effect on the cells. The cells in this image are very healthy looking.
So we're kind of bypassing the traditional route in a way, because we're able to just do a lot more tests a lot more quickly.
We're really trying to accelerate the process of drug development by making it a lot more efficient, and particularly by enabling researchers to understand how the human will respond to a drug, rather than using an animal as a surrogate for how a human will respond to a new medicine.
Under highly controlled lab conditions, the team are testing new drugs in the search for a cure to hepatitis B, a liver infection which kills 780,000 people annually. Hepatitis B naturally only affects humans and chimpanzees, making it very difficult for researchers to discover new medicines to treat the disease.
What we really want to be doing is to see whether you've got drugs that can actually cure things like hepatitis B. So how does that work in here? How can you look for that kind of thing?
We put the virus with the liver cells in the plates that we saw downstairs. The virus then will replicate within the cells, and we can measure how drugs affect the virus. We're really hoping there is going to be a breakthrough that will show the way to curing this virus, that will mean that patients will be able to take a treatment for maybe only a few weeks or months, rather than having to live on a lifetime of medication, and that's our goal.
As well as replicating individual organs in isolation, the next hope for researchers around the world is to create models of all the major organs and to link them together to examine how the whole body responds to drugs. The hope is that this global movement could speed up the search for treatments and cures for some of the biggest diseases facing humanity today.